High school biology lied to you. You see, in high school biology, whenever you were learning about DNA, you were probably taught that these were the only five nucleosides in the processes of transcription and translation, with adenine, guanine, cytosine, and thymine being a part of DNA, and adenine, guanine, cytosine, and uracil being a part of RNA. But have you ever heard of inosine? Inosine is what you get whenever an adenosine molecule goes through a deamination process in which it loses an amine group, also known as an NH2 or NH3 group, and replaces it with something else. In this case, in order for adenosine to turn into inosine, that NH2 group at the top gets replaced with an oxygen. And while not taking part in the transcription process in which DNA is transcribed to create mRNA or messenger RNA, inosine can be found in the translation process in which the mRNA that was created during the transcription process is then translated into amino acids. And during this process, inosine can either be found in the mRNA or in something called tRNA, also known as transfer RNA. tRNA being the thing that translates the code on the mRNA into amino acids. In the case of inosine on tRNA, the inosine, which can be identified by the blue eye right here, acts as something called a wobble base, which instead of pairing with only one nucleoside, like how G pairs with C and how A pairs with T or U, I can actually pair with three potential nucleosides, those being U, C, or A. Hence why it's called a wobble pair, as it's very versatile. And when the inosine resides on the mRNA instead of the tRNA, it basically does the same thing. It acts as a wobble base, as it can still pair with the nucleosides C, U, or A. It just gives some variability as to what tRNA can bind to it. Thank you so much for watching. If you like this video, you can follow me at Nelly the Science Geek on all platforms. And as always, stay geeky.